or not to take, what is the truth regarding the vaccines, and so on. So we're really delighted to be able to extend this platform to the general public, because we know there are a lot of you out there who have lots of questions that you would like to have answers to. And today we've put together a whole group of professionals who will seek to give you the information you need. We have eminent immunologists, we have clinicians who are actually dealing with the disease in their practice. We have public health specialists who are also involved in the rollout of the vaccines and so on. So fasten your seatbelts. We're going to have an exciting time, and I trust that by the end of this whole period, we will all have been better informed and able to thus inform others and educate others. So together we become each other's keepers, being our neighbors' keepers, as we are enjoined to by the good book. So I will hand over to my co-presenter, Dr. Bertha Sewa-Ayi, who is going to introduce the first speaker to you. Bertha, yours.
Okay, thank you so much, Prof. Professor Efo Hesit was one of my professors when I was a medical student. I looked at her in awe, and it's a real pleasure and an honor today as I sit here as a co-moderator with her. And the next person I'm about to introduce is a very eminent research fellow. And this morning she was telling me, you know, I didn't know Dr. Bertha went to Wesley Girls High School. And so this is a Wesley Girls High School Old Girls Association webinar. And it's interesting that Ivy did not know I went to her high school. And so networking is extremely important. And the Old Girls Association of Wesley Girls High School is very delighted to be able to present this. So Dr. Ivy Asantewa Asante is a research fellow at the University of Ghana Noguchi Memorial Institute for Medical Research. She obtained her PhD at the University of Hamburg, working from Heinrich Peter Institute of Eppendorf in Eppendorf, Hamburg, Germany. Her area of interest is emerging and re-emerging viruses, as well as respiratory infections. She's currently actively involved in supporting the Ghana Health Service with COVID-19 testing. There's a whole lot more I could say about her. I could probably write a book talking about her, but I think for the purposes of time, this should be more than adequate. And so Dr. Ivy Asante, the floor is yours.
Thank you very much, Dr. Bertha. So I will start now. Can you see my screen? Did I pause her? Please, can you see my screen?
Yes, we can see it, but it's not in presenter view.
All right.
Put it on slide view, yes.
Yes, okay. So I will talk about coronaviruses. So what kind of viruses are we dealing with when we talk about COVID-19? So these coronaviruses, they belong to a large family of viruses. These are known to cause potentially deadly diseases and also really very mild infections. So mainly respiratory, enteric, and hepatic. These viruses belong to the coronavirus group. And under this group, we have 4 different groups now. We have the alpha coronaviruses, we have the beta coronaviruses, and then gamma and then delta. The alpha and beta are known to cause disease in humans. So for beta coronaviruses, we have all these interesting ones like the SARS, which is severe acute respiratory syndrome virus, and also MERS. And now what we are dealing with, this is the SARS COVID-2, coronavirus 2. So like I said, they mainly affect cells of the upper respiratory tract, and in humans frequently cause mild to severe infections. And how are these viruses transmitted? So mainly through direct inhalation. So when we talk, when we cough, when we sneeze, when we have an infection and someone is close to us, the person can directly inhale it. And these droplets that come from when we are talking, coughing or sneezing, they can also land on surfaces. So like on our laptops or on our phones or whatever. And when we touch this and we touch our eyes and our nose and things, we can get it. There have been rarely cases of getting coronaviruses through fecal matter. So the viruses have also been isolated through feces, and mainly there's also spread from animals to humans. So where do these viruses come from? I've had this question several times. Where do these coronaviruses come from? So bats have been recognized as the natural reservoirs of a large variety of viruses, including coronaviruses. So these viruses circulate in bats without any problems. They don't give the bats any problems. It's when we keep disturbing their environment is when sometimes we get inoculated. So how does the virus get into us? It's mainly through intermediate hosts are known to play really a key role in coronavirus transmission to humans. So for example, for SARS coronaviruses, these SARS caused infections in 2003 in China. About 8,000 people were infected and 800 of them died. And it is known that the virus may have been transmitted through civet cats. And for MERS, since 2012, it has been causing an infection in the Middle East. And they are known to have been transmitted through young camels. So these are dromedary camels. Now, how did this SARS coronavirus 2 emerge? So this is the virus that causes the COVID-19 disease. How did this virus emerge now and giving us so many problems? So these viruses were known to have people who visited a market in Wuhan, China, were then diagnosed with this infection. And in this market, there were snakes, there are fishes, there are bats, or everything else is being sold on this market. So in earlier reports, implicated several organisms, implicated snakes, fishes, bats, either direct transmission or through these secondary hosts. But later through sequencing, it's been realized that the SARS coronavirus 2 shows about 96% similarity to coronaviruses in pangolins, these really scaly reptiles. But there is still not enough evidence to link SARS coronavirus to really anything. Therefore, the origin and then the emergence and transmission to humans remains largely unknown. So this is how the virus looks like. On the surface of the virus, we find these proteins we call spike proteins. So the virus uses this to infect cells. Then the genetic material is RNA. It comes in one long strand and it is really big, so up to about 32 kilobases. There's an envelope, and then there's also the nucleocapsid protein, which goes around the RNA. So when you look at this under an electron microscope, it looks like a crown. So like a crown that would be worn on your head. And this is how come the virus got its name. So when we stretch out this RNA, the genetic material of the virus, and we look at the functions of the various portions of this RNA, it looks something like this. There are like 6 large open reading frames. So it's the open reading frame that directs what should be synthesized, like what protein should be made. And the virus needs, to be able to hijack the system and then produce more of itself. So then there are about 14 to 16 proteins that are made, and there are the 4 major proteins: the spike, the envelope, the nucleocapsid, and then the membrane proteins. So how does the virus make more copies of itself? It needs to enter into the cell. So there are cell surface receptors which the virus prefers, and it binds to. these cell surface receptors with a spike protein. It enters into the cell, it releases its RNA genome, and then because the RNA is a positive sense, it's directly translated into the proteins that the virus needs. And then there is then replication. It makes more copies of itself. It assembles then into a mature viral particle, and this is then released from the cell. So this is how the replication happens. So at every point in this stage, the virus can change itself because it needs to adapt to wherever it is causing an infection. With adaptation comes changes, comes mutations. So these mutations can occur during the different stages. So when it is going to enter, when it is replicating, when there's going to be released, and especially during the replication stage. So the virus needs a particular protein to make more copies of itself. And this protein makes a lot of mistakes when it is replicating. It's an RNA, replicates really, really fast. So there's a lot of mistakes when the replication occurs. And therefore, there are several changes or mutations that then occur. So when we look at the picture on the left, this is a work that was done by Ngoy et al. In 2020. So it was with Noguchi and with Wagbib also. So the first sequence on the top, the first strain on the top, is the original Wuhan strain, and it shows in gray. And all the other sequences are from Ghana. And all these bars with different colors represent points at which there is changes in the virus. And you can see that even two viruses from Ghana don't look the same. You know, from different patients, they are very, very different. We see maybe one going through it, going along it. This is one particular mutation going through it. But then the viruses are not the same. They are quite different, you know, and we see this a lot for many viruses, especially for this coronavirus. So they produce things we call quasi-species, where the virus that enters is not exactly the same one that comes out, because it needs to adapt. It needs to adapt to the environment that it finds itself in. And the good news is that most of these mutations do not cause the properties of the virus to change. So if it can bind to a certain protein, it's still able to bind to that protein. However, sometimes there are a few major mutations or changes that can cause the properties of the virus to change. And these include things like increasing spread from person to person. So this is what we are seeing with the UK variant. Some of the variants or changes or may also cause severe disease, and some may also include immune evasion. So we've seen this recently. There's a collaboration, I think, between Wagbib, where Dr. Bediakun works, and then Yamachi, and then a different company. So this is like a dashboard where you can look at all the mutations that are happening. So currently, these are the variants that we see in Ghana. So we have mainly the variants from the UK, a lot of it here, very, very little South African, and then Algeria or Morocco, and then also variants from Nigeria. So this dashboard is interesting, and I've put the website here so that for people who want to see it, it's an interactive dashboard. So there are several things that you can see here. So for example, this B1 variant was first identified in Algeria and Morocco, and we have this in Ghana. So the website goes ahead to show the distribution across the country. So you can see wherever you are in Ghana, you can see exactly which variant is then dominating there. So these variants are then like, they have slight changes that have caused the virus to increase in transmission. So, and they are called, I mean, it's not a completely different virus. It's the same virus that looks slightly different. So I thought I'll show you some of the things we do here at Noguchi. So we collect samples, we do RNA extraction, and then we are able to diagnose this COVID-19. And then these are some of the labs that are currently testing for COVID in Ghana. So there are a whole lot more coming on board then. So what I'd like us to remember is that coronaviruses cause mild to severe respiratory diseases. Transmission is through direct inhalation of fomites. The natural reservoir is bats, and usually there's transmission through intermediary hosts. The origin of SARS is still largely unknown, and changes or mutations are constant. I mean, this is what we have to expect. Once there's a movement from person to person and from all over the place, and the virus keeps on moving, there will be lots of mutations, there will be lots of changes. Luckily for us, most of these changes will not affect the properties of the virus. There may be a few that might affect the properties of the virus, but most of them will not affect the properties of the virus. So once we keep on sequencing, we keep on looking at how the virus looks like, we'll definitely come across several mutations. All right, so I'm done. Thank you very much for listening. I will stop sharing my screen now.
Great, Dr. Aye.
Yes, I'm done.
Thank you very much. But Dr. Aye, you want to ask, go through a few myths with her? Thank you very much.
Okay.
So far, nobody has asked any specific question. So I think it will be appropriate if Dr. Manson goes through his, or was it Dr. Yao who was going to do his presentation next?
Yeah, we'll go first. But I just wanted to ask you,
Okay.
Ivy, so you keep saying we don't know where it came from and how it got from the bat to the humans.
Yeah.
So those who are saying it was manufactured, what is your impression about that?
So I usually tell people that I think we haven't gotten to the stage where we can manufacture such a virus. I mean, Dr. Bedako will talk because it keeps on changing all the time, you know, and there's lots of mutations. I don't think we haven't gotten there yet. We will not manufacture such a virus that will give us so many problems, and all of us are, a lot of people are dying. No, this virus is circulating in bats. We know coronaviruses circulate in bats, and it is because we ourselves keep on disturbing their environment. You know, wherever they are, we are going there, we are cutting down trees, we are breaking caves because we need places to live, you know, and we need to conquer everywhere. This is how come we have all these problems. Otherwise, they were fine in their bats and they won't give us any problems. Yeah. So no, we did not manufacture this virus. No.
Thank you very much. We'll come back to you again. Can I ask?
And Paul, can I just chip in real quick? The answer I usually give to those who talk about manufacturing is the fact that it wasn't until 1953 or 1963, I think it was 53, when Watson and Crick were able to identify or come up with the structure of the DNA that's, molecular engineering that has to do with the genetic material even started. But way before 1953, we had the 1918 pandemic. Yeah. We've had smallpox. We've had many things, many diseases that have occurred when human beings did not even know how to touch the DNA or RNA of any organism. So normally when I explain to people that this whole field of genetic engineering, and we're not saying it's impossible, we know that these days you can genetically modify, you can modify any DNA or RNA that you want, but just the fact that this technology is less than 50 years old, and before that was even discovered, we still had pandemics. And so I think it's important for people to realize that, otherwise, how would we explain the influenza pandemic? Were people also modifying, or were they manufacturing new viruses at that time? Probably not.
Great response as well. Thank you very much. We'll move on to our second speaker, And I'm delighted to introduce Dr. Yao Bediako. He is an immunologist, and he is a research fellow and the head of advancement at the West African Institute for Pathogens. It has a long name, WACBIP.
Yeah.
But his areas of interest are in studying the immune function among African populations as it's related to infectious and non-communicable disease entities, immunity, sorry. He's also interested in building sustainable research ecosystems in Africa. So Dr. Bediako, please, the floor is yours. Thank you.
Thank you very much, Professor. It's a pleasure to be invited to give a talk. Obviously, I did not attend Wesley Girls High School, but I think my strongest connection is that my father was head boy at Infectious. So I think there's my understanding is there's a good connection between Infectious and Wesley.
Yeah,
so,
yeah.
And I believe he used to say there were, in his time, sixth form ladies actually took their courses at Infectious, if I'm not wrong. So he has classmates who are actually Wesley Girls students. So they used to joke around a lot. I remember when I was growing up. So I have some connection to Wesley Girls. Yeah, I'm just going to share my screen. I'll try to walk through my job in this webinar is to talk about vaccines as the immunologist of the group. And even before that, I'll just chip in my own answer to the idea of these things being manufactured. Actually, work has been done to look, if you look at the structure of SARS-CoV-2, it's actually an imperfect virus. And a number of people have mentioned that if indeed it was actually to be manufactured, it would not be so imperfect. All the work suggests that it naturally evolved and it bears hallmarks of a naturally evolved virus. And I think we can be safe to say that if the Americans could blame the Chinese for manufacturing it, they would. The fact that they haven't been able to suggest that it most likely was not manufactured. Anyway, I'll stick to what I know, which is vaccines. So my talk is going to walk through 5 main points. I'll give you a brief history of vaccination, talk about what exactly a vaccine is, how is a vaccine developed. I'll talk specifically about the 3 main types of COVID-19 vaccines that are available. And then I'll end with a little bit of an overview of the current vaccine rollout. I think it fits in with talking about vaccine safety, to realize that this rollout has been going on for a number of months, and we have quite a bit of data on the safety of these vaccines. So vaccination is not anything new. I mean, I found during the discourse, people talking about COVID-19, it's as if vaccination just started. Vaccination goes back many hundreds of years. In fact, as early as 1000 AD, the Chinese were doing variolation, which is exposing people to a little bit of an infectious pathogen to induce what they hope would be a mild disease and then protect from more serious disease after. And this was born out of the realization that people often didn't get the same disease more than once. Variolation made its way to England in 1721. And then the first vaccination, however, happened in 1796. And this was done by a gentleman called Edward Jenner, who made an observation that milkmaids who milked cows would get a viral disease called cowpox, but they never got the much more severe disease, smallpox. And smallpox, many of us don't even know what it looks like. Smallpox was an incredibly debilitating and disease. Even if you survived it, you were left with terrible skin scars. And so what Jenner did was to intentionally expose a boy. The story goes that it was his servant's child, so we can debate the ethics of what he did, but he exposed this boy to cowpox, and then intentionally took material from the pustules of somebody with smallpox and exposed the boy to it, and the boy never developed smallpox. And so that is on record as the first vaccination. Actually, the word vaccine derives from the vaccinia virus, which is actually the virus for cowpox. So after Jenner, we have to wait a number of years before we actually have the first lab-generated vaccine. That happened in 1879 when Louis Pasteur developed a vaccine for chickens, for cholera in chickens. And then you had the BCG vaccine, which is a vaccine that is still in use today, being developed in 1921. Yellow fever vaccine, which everybody in Ghana, I think, is aware of. Certainly, if you've traveled anywhere, that yellow card you carry around, that vaccine was developed in 1938. And in many ways, the yellow fever vaccine that is in use today is almost identical to the original vaccine that was developed in 1938. So it shows how when a vaccine works and works well, it's almost lifelong in terms of its utility, provided the pathogen is still around. So yellow fever vaccine, we all know Noguchi Institute is named after a Japanese researcher who actually studied yellow fever, and I believe actually died from yellow fever, during his research. So, yellow fever now, we view it as something we get vaccinated against. I don't know the last time anybody heard of somebody dying of yellow fever. That is thanks to the vaccine. Oral polio is another one that we are all familiar with, the drops that are placed on the tongue of babies. We've seen that. I think anybody here who is a parent has taken their children to get vaccinated. In Ghana now, we're switching to injectable polio, so now it's a shot. But once again, these are vaccines, oral polio 1963. And I think Dr. Asante might give me the exact time, but I think last year we announced that, or it was maybe early this year, announced that we've eradicated wild polio completely from Ghana. That is a huge achievement and is due to vaccination. So I'll jump ahead. 2019 is probably the last time, before COVID, the last time we had a big breakthrough in vaccination was in 2019, where we developed the Ebola vaccine. We can all remember the concern about Ebola in West Africa. And within, I think, about a little over a year, the Ebola vaccine was developed. So that was actually, till we got to COVID, that was sort of the big win for vaccinology in more recent times. Hep B, 1986. And then, of course, 2020, SARS-CoV-2 raised its head. And by God's grace, after about a year of extremely hard work, we now have a number of vaccines on the market. But I show this slide to really drill home the point that vaccination is not new. Anybody here, probably every one of us listening, has received a vaccine in our lives, if not several. I remember when I was a child, we talked about the 6 killer diseases. Now in Ghana, I think the vaccination is up to 14 diseases that we give. And there are actually over 20 diseases that currently have vaccines. Now, vaccines, the reason we keep using vaccines is because they work. This is data from the United States showing what happens to diseases once you have a vaccine available. Diphtheria, polio, and measles are 3 diseases that we vaccinate babies against. And this data, the red dot indicates when the vaccine was introduced in the United States. And the line indicates the number of cases. And in this case, the scale is in the thousands. So when you see 200, that's not 200 cases, that's 200,000 cases. And you can see how rapidly these numbers decline once the vaccine is introduced. For diphtheria, it was 1921. For polio, it was 1955. For measles, it was 1963. And you can see how the line is at 0 now. And I think I was speaking to a good friend of mine who's a pediatrician, and she mentioned to me that she's actually never seen a case of diphtheria. And that is thanks to the fact that children in Ghana get diphtheria, a vaccination against diphtheria very soon after birth. Now, this insert shows what happens when we take our foot off the gas. This is again in the US. Now, you can see that since 1963, US recorded hardly any, if any, measles cases. But this insert is showing you cases reported in the year 2019. This is driven by the anti-vax movement, where parents who probably do not know what measles look like because they were vaccinated as children, decided in large numbers in the US, in states like Oregon and Washington State and other places, to not vaccinate their children. And so you've seen the emergence of measles outbreaks in a country like the US that has no business having measles, considering that these vaccinations are provided routinely. But I think this is a very powerful image to show that we can never get complacent till a disease is eradicated. And so far, smallpox is the only human disease that has been fully eradicated. These diseases still exist. And so vaccination is incredibly important. And so now in the US, you have well over 1,000 cases. I don't know what the current numbers are, but you have these pockets. And these are situations where children get very, very sick because of the, in my opinion, the irresponsibility of other parents. But vaccines save a lot of lives. They save, on average, estimated 3 million lives every year, and billions of dollars in healthcare costs that do not need to be paid. Now, bringing it home to Ghana, this is data from the extended program of immunization in Ghana. This began in 1978. So even in Ghana, I believe 1978 is the same year that Noguchi was founded, if I'm not wrong. But 1978 is quite, it's before I was born, I'll admit. Ghana has been vaccinated. So once again, even in Ghana, vaccination is nothing new. And you can see this is data similar to what I showed you for the US. This green line indicates the vaccination coverage, and you can see nicely that we're well over 90% coverage for measles vaccine in Ghana. And these are cases of measles in Ghana, reported cases of measles. And you can see how significantly they drop once the vaccine coverage passes a particular threshold. So vaccine preventable disease burden has declined significantly in Ghana. And it's great that we have such great coverage. In fact, this is slightly outdated. I think now the number of vaccines administered to individuals in Ghana as part of our EPI is actually 14. So I've talked a lot about how great vaccines are. Now maybe I'll tell you briefly about what a vaccine actually is. A vaccine is not really that complicated. In fact, I think many of the drugs we take are much more complicated than a vaccine. Vaccines are really simple biological preparations that are safe and effective and provide protection against harmful diseases. And the idea is they are preventative. So you administer it to somebody before they become exposed. When we talk about vaccines, you hear a lot of rhetoric about, oh, why don't you let my body's natural immunity handle the disease? The word natural these days is, in my opinion, completely misused. A vaccine is taking advantage of your body's natural defenses. A vaccine is simply doing what your body would do if it saw the disease, but it's doing so without you actually having to get sick first, because some of us may not survive the disease to build that immunity. And so a vaccine basically simply, in a sense, leapfrogs or provides your body's defenses with a little bit of a taste of a pathogen, allows your body's defenses to develop immunity or memory, and then that is what protects you from subsequent disease. So the vaccines are basically a way to train your natural defenses, but they do not cause the disease. In Ghana, there's a lot of talk about immune boosters. In my opinion, that's mostly hogwash. I don't really buy into that. Yes, a little bit of vitamin C is great. Most of us get enough of it probably in our diet. But people love the idea of immune booster. In my opinion, the best immune booster man has ever found or ever discovered is a vaccine, because it does exactly that. It boosts your immune system. Better than vitamin C, better than zinc, better than all the stuff that, Percocet or ginger, all the stuff that people are, drinking these days. A vaccine is the ultimate immune booster. Now, with COVID-19 vaccines, we've also heard a lot about concerns about the safety. So here I'm going to walk you through what happens when a vaccine is being developed. It starts off with preclinical work. Scientists like myself and Dr. Asante spend long hours in the lab trying to understand the pathogen. Virologists like Dr. Asante will try to grow the pathogen in the lab, in cells or in animals, and try to see how it behaves. Immunologists like myself will now try to look at how the immune system responds in these animals. Once we find a component of these pathogens that can induce an immune response, then that is administered to animals. The animals are challenged with the disease, and we see whether or not the animals are protected. Those are called preclinical trials. So it's preclinical testing. Once a candidate passes or succeeds in the preclinical testing, then it enters human trials, because obviously you cannot administer anything en masse to a group of people without having tested it. Phase 1 trials are purely about safety. You have healthy, typically 18 to maybe less than 30-year-old, very, very, the strongest in our population. These are typically college students who are enrolled into these trials voluntarily. Oftentimes, it comes with certain amounts of compensation. Kids in the US are always saying it's a good way to pay for your college. You just sign up for clinical trials. I had colleagues who did that when I was in school. Anyway, so safety trials are purely about safety. They give you the vaccine and they wait to see what happens to you. So they're looking for those adverse events that everybody is worried about. And so it's given to very few people, because obviously you don't want to, if there's something goes wrong, you don't want it to affect too many people. Once a vaccine makes it through that safety assessment, it enters a phase 2 trial. Now, at this point, you're testing in hundreds of people. Before it was tens, so maybe 20, 30, 40, 50 people at most. Phase 2, now you're in hundreds of people. Again, assessing safety because we all vary genetically, and maybe that initial 50 people was not very representative of general population. So now you go into a larger population of people. But here you're also beginning to look to see what happens to the immune system. So they'll probably take some blood samples, look to see if you're getting an immune response. But you're not really looking to see if the vaccine is working. In this case, once again, is your primary concern is the vaccine safe? Once it passes that second phase, now you enter phase 3. And this is really the do or die for the vaccine. Does the vaccine actually work? Now, at this point, you're vaccinating thousands of people. For the COVID-19 vaccine, the Astrazeneca one, for instance, there was over 20,000 people in 3 different countries, the UK, Brazil, and in South Africa. So now you're going in different geographic regions. You're trying to get a broad spectrum of people. And now you're administering the vaccine to thousands of people. Now here, these are people who are potentially exposed to the disease. And so in addition to watching to see if there's any negative reactions, now you're also going to count how many people who you vaccinated actually get the disease and how many people who you haven't vaccinated also get the disease, because now you'd have, typically it's a blinded trial, so people don't really know what they've got. They've either gotten the experimental vaccine or they've gotten a placebo, which might be a rabies vaccine or some other vaccine that everybody knows works, but it's not specific for the pathogen of interest. And then basically you're comparing numbers of infections in your 2 groups. And that's how you determine whether or not the vaccine is effective or efficacious. So once the vaccine is only going to be regulated, only going to be approved for mass distribution once it has passed these 3 phases. And even then, once it enters limited use or emergency use authorization, as has happened with COVID-19, these vaccines are constantly being monitored to catch any negative events. So here I'm going to talk briefly about the types of COVID vaccine. There are a number of ways to make a vaccine. A vaccine is all vaccines are trying to induce an immune response, but they can do it in different ways. There are different sort of technologies, you could call it, that have been used. The original vaccine, Jenner's vaccine, was he took a pathogen that was related but not identical to smallpox and injected that into a child. Other vaccines have taken the actual disease-causing organism, weakened it in some way, so either killed it by heat or some other method or chemicals, or weakened it such that it cannot replicate, it cannot grow, and then inject that into individuals. And then the idea is you induce an immune response without the disease. As technology has evolved, we've moved away from live vaccines because, especially with the disease-causing organism, because in people who are immunocompromised or others, that can be problematic. We see this sometimes with the oral polio, which is why we've discontinued that and we're using the injectable polio. Live vaccines have worked really well, but now we have capacity to do things slightly differently and in some ways a safer way. So the first type of vaccine that we're going to talk about with COVID-19 is called a viral vector, a recombinant viral vector vaccine. In this case, you are delivering genetic material from your pathogen of interest, but it's not in the actual pathogen. It's in a related or a harmless virus. So in this case, you're taking an adenovirus. Adenoviruses cause colds. In the case of Astrazeneca, it's actually from a chimpanzee. In the case of Sputnik, I think it's a human adenovirus. But these are viruses that cause hardly any disease. They're harmless. But you've genetically modified them, so we call them recombinant, and you've placed in them DNA that would encode for a protein from coronavirus. So in this case, the spike protein that Dr. Asante mentioned. So this recombinant virus, it cannot replicate. So even the adenovirus cannot replicate. It has been modified such that it just enters and it will flush out. It cannot divide and grow. And then secondly, it is only expressing a piece of SARS-CoV-2. So the Ebola trials that were aborted in Ghana, in my opinion, were aborted because of a misunderstanding. The Ebola vaccine that was brought to Ghana was a recombinant like this. It was not going to give anybody Ebola. It was only expressing a piece of Ebola. But I think, public misinformation, people got worried and it was aborted, which is unfortunate because then other countries got to take advantage of the trial that we did. But anyway, in this case, the first type of vaccine, so AstraZeneca, which is the vaccine that has been in use in Ghana that I have received, I'm sure Dr. Asante received, probably many people listening have received, is a recombinant, a chimp adenoviral recombinant virus that is expressing spike protein. The second type now says, okay, we don't want to put a whole virus into somebody. Instead, we're just going to put a part. So it's a subunit vaccine. It may be a protein. So in this case, you might just put the spike protein into the vaccine, inject that into somebody, and then try to induce an immune response. Novavax, which is made by Novartis, it is currently in phase 3 trials, is such a vaccine. If that becomes approved, it may become available in Ghana. So it's important to understand what that is. The final type, and the most controversial, it would seem, is the RNA viruses, or the RNA vaccines, or the genetic material vaccines. In this case, you've stripped away, instead of using another virus to deliver your payload, you are now just putting in some RNA that encodes for the spike protein in this case. It's typically in a lipid, so it's in a liposome, so it's in the little nano part, like a tiny lipid capsule you could think of, and that is injected into people. Now, this represents, probably you could call it, the most advanced or the most cutting edge or the most recent technology in vaccinology. But it's not that new. It's been around for 10 years. It's just that it was still somewhat in development. And that is purely driven by finances, by resources. So it is true that the Moderna and Pfizer vaccines are the first human vaccines licensed using this RNA technology. But this has been in development for a very long time, and in many ways represents an even safer way of administering a vaccine. It's not another virus. RNA is very unstable. So once it gets in, it does what it's supposed to do and it's cleared. And also, it doesn't induce as much of an immune response. Many of us who got the adenovirus were down for about 24 hours. I mean, it hit me pretty hard, I must say. My friends were laughing at me that I was on TV saying there's no side effects, and I couldn't even move after I had it for a day. But I had fever and I was shivering, but I was not in any danger. It was just my immune system responding to the vaccine. The Pfizer and Moderna vaccines are designed to actually have fewer of these sort of feverish side effects because it's just a bit of RNA. They don't induce the same punchy immune response as you see with the chimpadino. So I hope that gives you an idea. I'm happy to answer any questions as we go, but these are the three broad technologies. And in many ways, they're all achieving the same thing, just in slightly different ways. But the main implication is cost. Moderna and Pfizer cost a lot more than the Astrazeneca vaccine. And also, because it's RNA and it's somewhat unstable, you may have heard the idea of cold chain. They need to be stored at cooler temperatures. And in our setting, as a low middle income country, where things are going out, it may be okay in Accra, but when you start going to rural areas, you're transporting these vaccines, you may not have liquid nitrogen or a -70 freezer, and the vaccines may go bad. So having said that, Pfizer, I believe, has said that actually our vaccine is fine in a normal freezer. They've done some work, so they're trying to get approval for it not to be required this ultra low cold temperature. So we may be able to use that in Ghana more easily. But anyway, those are the differences. Quickly, there are lots of vaccines in the pipeline. COVID has been a boon for vaccinology because it's a global disease. You've got the whole world to vaccinate. So there's almost an ending market. No one company can supply the demand. And so lots of companies are investing in this. And that is another reason why we've done this quickly. There is a clear demand for it. Countries are willing to pay whatever they can for these vaccines. And so there's great incentive for companies who ordinarily would not really care about vaccines to develop vaccines. And that is partly also why we have so many vaccines in the pipeline. Finally, how are we doing? I keep telling people, Ghana has so far received 600,000 vaccines. The rest of the world, 420 million doses have been administered. Ghana, we were sort of back in the pack. The US has done 118 doses. I'm sure Dr. Ayi has got hers a long time ago. China, 64.9 million. India, 42 million. United Kingdom, 28 million. Ghana, maybe 400,000. I mean, that is how far back we are. But the reason these countries are doing this is because, as Dr. Asante showed you, the virus is constantly mutating. These vaccines were developed last year with spike protein from the original Wuhan strain. The virus is constantly mutating. The fear is that you will get an emergence of a variant that is sufficiently different from the original strain that the vaccine will no longer work. And so countries are trying to vaccinate as quickly as they can to get rid of the virus from their population before it has time to mutate. Israel has taken the lead. I mean, Israel, smaller population obviously than the UK and the US, but has already done well over 60%. I think this number is actually a little bit old. It's about well over 60% of their population. Israel was willing to pay way above market. I think they paid $40 per vaccine to Pfizer to get ahead of the queue. And so they've gone all out to vaccinate as many people as they can, and they will probably soon hit herd immunity as far as the vaccination is concerned. But it's been worth it. Data they've released from a study shows that people who are vaccinated, 600,000 vaccinated people, had 94% decline in symptomatic cases of COVID-19 compared to people who were not vaccinated. So I would say the investment has been worth it. And countries like ours, though we don't have the same resources, we cannot be wasting the vaccines that we actually have. And so when vaccines arrive, the hope is that when it's your turn and you are called, people will not be hesitant and people will be rushing to try to get vaccinated. With that, I'll just like to say thank you for your attention. I'm happy to take any questions. Yeah, thank you again for inviting me.
Thank you so very much, Yao. Wow, you've taught us quite a lot. There are a few questions for you.
Sure.
When you were talking about the mRNA, a question that people are most afraid of is that this RNA can somehow get into our DNA and change our composition forever. They talk about it being like a chip which can be manipulated from outside. What are the effects? What could be the effects in the long run of any of these vaccines you have talked about?
Thank you. First of all, RNA does not integrate into DNA. That is biologically impossible. So I think the first thing from a biology standpoint is that, the fact that this is RNA doesn't typically enter the nucleus. It's found in the cytoplasm. It's bound by ribosomes. They make proteins. RNA is degraded. So this RNA is likely not going to hang around very long. And it's highly unlikely that it's going to get into the nucleus. And then, just the idea of it modifying your genes is very unlikely. And also, it's not being injected into your bone marrow or into your stem cells. It's going into your arm. It's going to go into your blood. It's going to enter probably dendritic cells that are going to, make spike protein. The RNA will be transcribed into spike protein. The dendritic cells will present this spike protein. You'll get T cells recognizing it, and you make T cells that will recognize spike. From a biological perspective, those dendritic cells will then die. So even the cells that have been modified are going to be lost. So probably within a week, 2 weeks, the only thing left of the original vaccine will just be the effect of now you have T cells that can remember, or you may have some B cells that can make antibodies to the spike protein, which is what you want. Vaccines typically don't have lasting, people are talking about, I want to wait for other people to get it, and after a year, I'll also get the vaccine. Drugs can have long-term side effects. They are chemicals. Biologics like vaccines actually typically don't. Majority of vaccines, if something bad is going to happen, it's going to happen in the first 24 to 48 hours after you get it. It's very unlikely that a vaccine is going to have long-term ramifications. Now, contrast that with COVID-19. There's a growing body of evidence that long COVID is pretty bad. And so, when we talk about vaccines, also a risk benefit. If COVID-19 wasn't that bad of a disease, we wouldn't bother with the vaccination. There's no vaccine for the common cold because, well, it doesn't really kill anybody. It just, affects your productivity at work, makes you feel a little bit unwell. But there's no vaccine for it because, hey, it doesn't kill anybody. COVID kills, and COVID can have long-term effects. So yes, there is the idea. The RNA vaccines can be modified, but they are modified later on. So right now we heard about South Africa having a variant that was, that could evade the Astrazeneca vaccine. I believe Moderna have created a new vaccine that specifically targets the South African variant. Now that is the power of the RNA approach. It's very easy to sort of reprogram, not in a person, but you can redesign your vaccine. RNA can be synthesized very easily, and you just repackage it and you make a new vaccine very quickly. I believe AstraZeneca have also tried to make a booster to specifically take care of that problem. But yes, there is no risk, in my opinion, and I think most immunologists and cell biologists will bear me out. that there's really no risk of any genetic changes as a result of an RNA vaccine.
Thank you. Another question has just come up. It says, has the Astrazeneca vaccine gone through the phase 3 testing, or is it still in the phase 3 testing? The use of this term experimental is worrying a lot of people. So they feel we're being used as guinea pigs.
So no, this is not an experimental vaccine. This AstraZeneca vaccine went through phase 3 trials in the UK, in Brazil, and in South Africa. Altogether, I think well over 20,000 people were involved in that phase 3 trial. So AstraZeneca has completed phase 3. Our FDA would not have approved it if it had not gone through phase 3. You cannot approve a vaccine. No country's FDA is going to approve a vaccine without seeing phase 3 trial data. Moderna and Pfizer completed phase 3. Sputnik 5 has completed phase 3. Johnson & Johnson, I think, has recently completed phase 3. Novavax is still in phase 3, which is why it's not approved. So these are not experimental vaccines. But having said that, I also want to, I want to use this opportunity that we should not be afraid of clinical trials, though. It is a shame that Ghana was not one of the sites for the AstraZeneca trial. South Africa was, and so they got AstraZeneca vaccine very early. And actually, they were able to determine that, oops, AstraZeneca doesn't work too well for us because of the variants we have. We have no idea. We are, thankfully, our dominant variant, as Dr. Asante showed in her slides, currently is B117, which is a UK variant. UK have studied it and showed that AstraZeneca still works for it. We have other variants in this country that no one is studying because they don't have. But because we don't do trials here, we actually are running blind. We are just praying that the vaccine will work. Thankfully, right now, most of our virus looks like the UK one. So we are just copying what they do. But if we saw an emergence of a uniquely Ghanaian variant, we would have no capacity to actually assess how good the vaccine works because we don't take part in clinical trials. So I think this is something that I just use the opportunity to try to inform people. There's nothing about being a guinea pig. Clinical trials are very, very carefully regulated. In fact, right now, Noguchi is carrying out the Lassa fever vaccine trial. Clinical trials are crucial to vaccine development. We cannot say we want to become a vaccine developing country and say we don't do clinical trials. I mean, I say, who should test it for you to use? Right now, we are benefiting off of the people who volunteered to take part in the trials. It is in my opinion, it's a responsibility. It doesn't mean we should allow ourselves to be exploited. But clinical trials are very tightly regulated. If they are with reputable companies, with the WHO's approval, they are things we should be welcoming. We have the capacity in this country, the brains in this country to run really great clinical trials. We are politically stable, we're economically stable. We would be a great place in West Africa. I can't think of anybody better in West Africa to be the hub for clinical trials. And that would really attract investment into our research ecosystem. But because we have this mindset of we don't like clinical trials, unfortunately, it stands in our way. But anyway, in this case, the vaccines in Ghana are not experimental. They are fully regulated, fully phase 3 approved. And so it's not a clinical trial. It's, at most it's a phase 4 implementation. So we are still collecting data, but they've gone through.
Okay. Thank you very much. Nana Nyako, I hope that answers your question. Irabna Akiwumi is asking about the AstraZeneca blood clot, but I'll keep that and get to Rada when, as it's her turn. Thank you very much, Dr. Yabediakun. We really have been educated about vaccines, what they can do and what they cannot do. So we shouldn't keep going wild with these allegations. I'll hand over to Bertha to introduce Dr. Rada Hackman. Thank you. Have we lost Dr. Ayi?
No, I'm right here. I'm right here. Thank you very much for that presentation. That was an excellent presentation. So I have the pleasure of introducing Dr. Hackman. And before I do that, I'll just reiterate once again that thank you for joining the Wesley Girls High School Old Girls Association webinar on COVID-19 myths. And just for your information, Professor Ifwa Hesse is in the 1969 year group. I'm in the 1987 year group. And Dr. Ivy, who spoke earlier, is in the 1997 year group. And the next person I'm about to introduce is a proud member of the 1976 year group of Wesley Girls High School. So Dr. Rada Hackman, born Dua, is a general practitioner and medical director of GIA Clinic in Accra. She graduated in 1986 from the University of Ghana Medical School after completing Wesley Girls in 1976. She's been in private practice for a whopping 33 years since 1988. She's been involved in testing for COVID-19 disease since January 2020 and treatment of COVID-19 cases since March of 2020. She's a member of the National Medicine Selection Committee of Experts, which was set up to develop the standard treatment guidelines for COVID-19. She's a member of the Ghana Medical Association Infectious Disease Committee and also a member of the board of Kolebu Teaching Hospital. She's appeared on television many times to talk about COVID-19, and I think she's going to greatly enlighten us about this disease from the Ghana perspective. And so with great pleasure, please welcome Dr. Rada Hackman. Thank you.
Thank you, Dr. Bertha. Hello, everybody. Yeah, I was done such a great job. I don't think I need, or anybody needs any more convincing. But I'm going to try and take you through the COVID pandemic 2020 through my eyes, through my consulting room. So hang on, let's just get started. So, okay, so the background to this, as we all know, we woke up on the 1st of January 2020 to the news that China had reported a very strange pneumonia in Wuhan, which was unlike anything they'd ever seen before in terms of the transmission and the mortality, morbidity and mortality. So here in Ghana, we got working. We had our case definition. We were looking at cases, anybody who had influenza symptoms and a temperature of 38 and above was swabbed and the samples taken to Noguchi. My facility has a long relationship with Noguchi spanning about 10 years, thanks to influenza. So as soon as Noguchi alerted us they had the reagents for testing for COVID, we started sampling patients that fitted the case definition. And we started this, I think, towards the end of January or the beginning of February. So here in Ghana, we diagnosed our first 2 cases on March the 12th, which was the day after WHO had declared COVID-19 a pandemic. The disease, as we all know now, was caused by the SARS-CoV-2 virus. And like I said before, we had been told what the symptoms were. We're supposed to look out for patients who had fevers and who had flu symptoms. So in my clinic, just like any other facility, I'm sure, the protocol was for patients who came to the clinic who had flu symptoms to wear masks. We'd actually offer them masks when they came into the clinic if they had symptoms. Everybody else, was fine. Which brings me to the asymptomatic COVID patients. We found this out in our facility, it was quite interesting because in March, after we diagnosed the first few patients, we went into lockdown. And whilst we were in lockdown, we were vaccinated, we were asked to test travelers who had come into the country from the 3rd of March all the way through until the point of lockdown when our airports were closed. And we actually in our clinic got our first positive COVID patient about maybe 10 days after Ghana had diagnosed the first 2. We were now vaccinating a lot of travelers, people who had come, who didn't have any symptoms or maybe had mild symptoms. And to our surprise, most of the patients who tested positive were quite different from the case definition we had been given. Most of them did not have any temperatures at all. And most of them actually did not have any symptoms. They had no respiratory tract symptoms. That made nonsense of the protocols we had, which was just giving masks to people who had symptoms. So we very quickly realized that maybe everybody who came into the clinic should wear masks. And we actually started doing that, but it didn't occur to us that we should be masked, as well. That, of course, came later. So basically, we realized that we had a whole lot of patients who were positive, who were testing positive for the virus, who either did not have any symptoms at all or had very mild symptoms. We also realized during this time that we were doing, this expanded testing. We're testing contacts of the positive. Of course, every time we had a positive case, we would test the contacts of these patients. And we realized that there was actually quite a number of patients who were positive but did not appear to be sick. They did not look sick. The asymptomatic and the mild patients were, played quite a bit of a role in community spread. So what are the symptoms as we know them now for COVID-19? In adults, as we know, they may be asymptomatic. They may have a fever. Very few actually have those really, really high temperatures. They usually may be very mild temperatures, 37, 37.4, 37.8. Some have coughs, but not all of them have respiratory tract symptoms. Sometimes they come and the only thing they complain about are headaches and then body pains. They complain about fatigue. So you need a really, really high index of suspicion to test some of these people. There was one that came up, which everybody sadly identified with, the reduced or loss of taste or smell. That was actually bringing a lot of patients to the clinic. Once they woke up in the morning and they couldn't smell their soap, or they couldn't smell their breakfast or whatever, they knew there was a problem. And sometimes that was the only symptom they'd come with. There were patients who would just come because they lost their appetite. There are patients who would come with just diarrhea and vomiting. And a lot of these symptoms, I'm sure you all know, are like symptoms of most other diseases like malaria, maybe even typhoid or whatever. So sometimes people, would treat themselves for malaria or whatever before they come. In children, same thing. They may be asymptomatic. They may come with a skin rash, pink eye, maybe just a tummy ache, or maybe a child just doesn't feel well. They're irritable, they're drowsy, they have a runny nose. They may not have a runny nose. They may not have any respiratory tract symptoms at all, again. So just so that you know, yes, children also get COVID. It's usually mild. They're not usually as sick as the adults. I haven't seen a child as sick as an adult yet. So clinical presentation, I think we've more or less talked about that. Yes, they may come with no symptoms at all. And we normally pick these up, as I've said, as I've already said, through contact tracing. That's how we pick up the asymptomatics or people who just walk in to test because they're wondering what their status is. The illness may range from mild to severe. In the elderly and immunosuppressed, you may not get the classic symptoms that I mentioned before. They come with very atypical symptoms. Maybe an elderly person who's just gone off her food, sleeping a bit too much, just generally unwell. So what's a mild case of COVID? They're usually those who present with mild cases of the symptoms, mild symptoms, as I've gone through already. With severe symptoms, the really, really severe symptoms may actually have pneumonia, just pneumonia, which may not require them to need, to be given oxygen. But then you could also have, patients with very severe pneumonia. These require oxygen. You could have patients who have, an acute respiratory distress. You could have patients, with blood infection, sepsis, septic shock. These are a little technical, but these are just examples of what severe patients present like. So patients will typically present with very mild symptoms. How can you tell when they're going to go from mild to severe? What makes a person more likely to progress to severe? Before we get there, what investigations do we do for patients who come with symptoms, like I've said? You've all heard about the PCR test. It's a household name now. Now, the PCR test, just like Dr. Ivy has explained, We would normally take swabs, from the throat, deep down in the throat. We take swabs from the nose. I remember there was a time, I think, was it June, July, August? Ivy might correct me, where we actually ran out of swabs. And for the most part, a lot of testing centers were using expectorated sputum, which is basically phlegm that you've coughed up from your chest. You can imagine how messy that was. There was the other one I forgot to write down, which is nasal wash. So you push in a syringe a bit of saline into one nostril, then would come out through the other nostril. You collect that in a tube, and you send that to the lab as well. In those days, Noguchi was the main place we're sending samples to. In some cases, we do blood tests, and these are usually for people who we consider high risk. And these are patients who have other conditions. I'm sure you're now familiar with the term underlying medical or health conditions. So these are people who are elderly, maybe over 60, people who have high blood pressure, people who are diabetic, asthmatic. Sometimes we would want to do blood tests for these people just to establish, a baseline so we can monitor them. And these are generally, what we call a full blood count test. We would check their kidneys, we check their liver. The D-dimer test is just a test to help us know whether the patient is, likely to form clots or not. The inflammatory markers, these are, again, tests that help us to know if this patient is progressing from mild to more severe forms. Then, of course, there's a chest X-rays, CT scan, ECG. These are done usually for more severe disease. So these are just pictures of my staff. The top one is in the early days, so we tried a drive-through. I must say it was more of a drive-in and then reverse. Windows are rolled with stops, and then they drive out again. And the lower one was when we had a lot more people to test, and we'd find a site, an off-site from the clinic, and do the same. Prevention and control of COVID. I'm sure we all know that until the advent of vaccines, there were no proven medications for the prevention of COVID-19. So this is just to bust another myth that, sometimes when patients test positive and you tell them, the first thing, oh, but doctor, I was following all the protocols, I was doing everything. What exactly were you doing? Oh, every day, twice a day, I was inhaling neem. I was, drinking lemon, ginger. I was doing all the right things. I was wearing my mask. I wouldn't take it off. I was washing my hands. I was doing everything. There really is, until the advent of vaccines, there was no medication which was proven to prevent COVID-19. Ivermectin inclusive. It's not proven that it can prevent you from getting COVID. On the left, for anybody who's in Ghana knows, the picture on the left, that's a Veronica bucket. And this is actually just outside our front door in the clinic. It's been wonderful. You fill the bucket, on top with water, you wash your hands. This is pedal operated. You wipe your hands with tissue and you discard it. So that's a hand washing. We're all very familiar with sanitizers in various forms, the gels, the sprays, We've talked about mask wearing, of course. By this point, we all knew that everybody should wear a mask. The patient should wear a mask, the health worker should wear a mask as well. Distancing, we all know about that as well. Avoiding, crowded spaces, and these are proven to prevent the spread of COVID. Vaccines are like the icing on the cake. That's proven to prevent severe COVID, and also they're proven to prevent death. In February 2021, Ghana's FDA approved 2 COVID vaccines for emergency use. This is the Astrazeneca vaccine and then the Sputnik from Russia. How have we treated COVID? The early quarantine of suspected cases, you all know, psychosocial support, physiotherapy for those who, need help coughing up their phlegm. When we had our first wave, this was March, April, May, June, July, almost all positive cases were admitted in isolation centers or treatment centers. So if you tested positive and you couldn't isolate safely at home, you were put in an isolation center. I'm sure you all remember the Pentecost church facility, which could take, I think, about 800 people. So these were patients who were either asymptomatic, they had mild symptoms, but they couldn't isolate safely at home. They were sent to the isolation center. Those who were a little bit sicker were taken to any of our treatment centers. In those days, we didn't have too many severe critical cases, but our treatment centers got full anyway because we're admitting most patients and treating them. The second wave happened, and it started actually towards the end of December. And this was totally different from the first wave. People were a lot sicker. We had more severe cases, and it seemed to be transmitting a lot, faster. With the second wave, we saw a lot more younger people. With the first wave, I didn't see too many ill younger people, but with the second wave, we saw a lot of younger people who were actually quite ill. Some of them ill enough to be admitted, and treated. Especially with the second wave, we did a lot more home-based care for mild and moderate cases, and these were done under the supervision of districts, COVID teams. Every district has a COVID team made up of doctors, nurses, sometimes even a psychologist, a nutritionist, and everything. And these were directly in charge of taking care of the home-based patients. Sometimes they got a little overwhelmed, and doctors like me, from the comfort of my home, would also help as well. We encourage these patients to spend a lot of time outdoors, get a lot of physical exercise, good nutrition, hydration, good ventilation. And for those who had bad coughs and were chesty, we encourage them to use humidified air, steam, and inhalation. We also monitored them, at home, because we needed to know, as soon as it was, they were changing from mild, moderate to severe. And we noticed that this frequently happened in the second week of the illness. The first week was usually, not too bad. With drug treatment, again, as you all know or should know, there's no proven medication that can cure COVID-19. The treatments we gave depended on the clinical presentation, as in if they were mild, moderate, or severe. This is a pulse oximeter, and this is something I encouraged all my patients to get. And certainly with our patients that I had, I actually bought, a stash of pulse oximeters, which I would lend to them. And these are actually readings from actual patients. So twice a day, they'd send me a picture via WhatsApp so that I'd know how they were doing. So a pulse oxygen, the one on the left shows an oxygen saturation of 99, which is good, and a pulse of 77. The one on the right, as you can see, shows an oxygen saturation of 94. This shows that you're heading into trouble. And at that point, you need to start making preparations to get that patient into a treatment center. All severe cases, must be admitted and treated at treatment centers. Like I said, there's actually no medicine that has been proven to cure COVID, but there are medicines which have been found useful for preventing complications of COVID-19. These are the complications that usually lead to death. And we are talking about things like the cytokine release syndrome, acute respiratory distress syndrome. This is when the patient really, is in actual distress and can't breathe and needs some support. There are also medications which can prevent the formation of blood clots. All the other treatments we give is supportive, just to help the patient's, immune system fight, the disease. And so such a time as they're well and able to go home. We have a number of treatment centers, but the Ga East Hospital is one of our main ones because that can take, it has 100 beds. It can take 100 patients. And then there's a second one, the Ghana Infectious Diseases Center, which is actually new and was built just last year. I think it was commissioned in September, at a time when our numbers were actually going down. So for those of you who don't live in Accra and haven't had the benefit of seeing our purpose-built infectious disease center, this is what it looks like. It's very nice, very purpose-built. Even the air circulation, it's comparable to any infectious disease center you'll find anywhere in the world. The flooring, the walls, the paint, the texturing, the airflow, everything is, state of the art. So I thought I'd talk a little bit about the post-acute COVID syndrome, because sometimes when you recover from COVID and you test negative, it doesn't end there. Sometimes symptoms can persist long after testing negative. Sometimes patients actually get better. There's an initial improvement. And then suddenly they start developing symptoms such as fatigue, weakness, shortness of breath. And you can't really predict who will, go on to have post-COVID syndrome. Patients who are mildly ill, even patients who are asymptomatic, can end up with a post-acute COVID syndrome. And it's quite distressing for patients. And I'm seeing a lot of that now. I'm sure there are any clinicians on here, they're seeing that as well. There's really no specific treatment, and you can't predict how long it's going to last. Some patients have felt unwell, for 3 months after they had COVID, some for even longer. They keep coming to the clinic, I feel tired. There isn't so much you can do for them, but just to treat whatever symptoms they have and just assure them that eventually it will go. Okay, so these are the common symptoms of long COVID. Sorry, the post-acute COVID syndrome is also called, long COVID. Some call them the long haulers. And these are some of the symptoms they complain about. It's almost like they've got COVID all over again. And in fact, some come back because they say, we feel like we've got COVID again. But this time you test them and the test is negative. So there's fatigue, shortness of breath, blah, blah, blah. Some of them have bad dreams. Some, have difficulty sleeping, especially those who've spent time in the ICU. I think that's quite a traumatic, time for patients who are severely ill. They have flashbacks, some are depressed, they feel anxious. They talk about, some people talk about memory fog, just problems with concentration, problems with memory. They complain about palpitations. There's some who actually have damage to vital organs such as the lungs, the heart, the kidneys, the liver, and the brain. So you have to keep monitoring such patients to pick up damage to these organs and then deal with them appropriately. Some may have heart attacks, strokes, pulmonary embolism, blah, blah, blah. I must say that it's been said, some patients who have post-COVID syndrome, who've had vaccinations, for reasons we cannot explain, suddenly begin to feel better. So that's, another reason why you might want to consider getting vaccinated if you've had COVID already. Then reinfection. And this is where a person can recover after an initial infection and then later become infected again. I've seen 3 of my patients who, funny enough, I saw them all, 2 in December and 1 in January. And these are patients who had actually tested the first time, come to me the first time with symptoms, had been found to be positive. And the first one I saw had had COVID in June. It's a young guy, actually. I think he was 24 years old. And then he came back again in December, was between Christmas and the New Year, with symptoms again. And I tested him, we swabbed him, and he came back, positive. Soon after that, we had our second and we had our third. So this is just so you know that, having COVID once can sometimes, not just be the end of it. You may have long COVID, you may also get, reinfected. And I think these are reasons for, to convince you to try and get vaccinated. I think Dr. Yao talked about natural immunity or vaccine, because sometimes patients say, I'm not sure about the vaccine. Isn't it better if I just, got the disease? That's natural immunity. Isn't that better for me? But the thing is that it's very difficult to predict what the cause of a natural infection, could be. So choosing the disease over the vaccine may not be wise. From what I've said, you could be asymptomatic. Good for you. You may have mild disease. Good for you. But the reason why we're all wearing masks and keeping away from each other and living this very unnatural life is because we are afraid of getting the more severe forms of COVID. We're afraid of probably even dying from COVID. So a vaccine is predictable and safe. And it's been proven to prevent severe disease and death. So I have seen what COVID can do. I have seen the damage that COVID can do. I've seen the second wave in Ghana. I've seen what it did to people. I saw people who came close to death, but thanks, God, they recovered and came back, home. Would I take the vaccine if I was offered a vaccine? The answer is a very big yes. I have a fear of needles. I got vaccinated. Thank you very much for listening.
Thank you very much, Radha. That was President. I think there are a few questions that in the chat.
Prof, I missed, if you said anything, yes. I missed it.
Okay, no, I was just saying, people were asking about any of your patients suffer any, and what about this issue of blood?
Sorry, Prof, I can't hear you. No, I can't.
Hello?
It's breaking so badly. Hello?
Can you hear me now?
It's breaking. I can hear
you.
Can
you hear me now?
But yes, I can.
All right. Okay. Is it better now? Yeah. I was saying that there were some questions about causing severe allergic reactions. And if any of your patients
about the vaccine
developed such severe reactions that they had to get it? Yes, of the vaccine.
To the vaccine.
And
no.
This brouhaha about, yes, or this about clots caused by blood caused by Astrazeneca vaccines. Would you like to comment on those?
Yes, I haven't seen any severe reactions to the vaccine in the clinic. What I have seen, I've seen, well, 2 patients have come to see me with high temperatures, and these are temperatures of over 38. That was just two patients actually came to see me. And we just gave them paracetamol. One had ibuprofen, the other just paracetamol, and she was fine. Most people who called me just complained about, body pains and feeling weak, which really went away after a couple of days. I myself, hello?
Yeah, we can hear you.
Oh, okay, sorry.
We can hear you.
I myself felt absolutely nothing. I felt, it wasn't even painful. After all my drama, it wasn't even painful at all. and I didn't have any side effects, at all. With regards to clots, deep vein thrombosis or clots are things that happen anyway. They are not uncommon. And it's so funny because this year in January and in February, I've had 2 patients who've had, blood clots, just to show you that it's nothing out of the ordinary. None of them had COVID before, and none of them had been vaccinated. The second one was actually, I think, 2 or 3 days after we started vaccinations, in Accra, and he came to see me with a swollen leg, which he'd had for about 5 weeks. So clots happen, and when you're vaccinating a large group of people, you're vaccinating people who have comorbidity. People have other conditions which would make them prone to these things anyway, whether they've been vaccinated or not. So for example, these 2 people I saw, had they been vaccinated, one would have thought, this is related to the vaccine. But both had conditions which made them more prone to, to forming clots. Plus, I think it's been investigated in Europe, and I think we all know what, the result has been. I haven't had a patient yet, and I haven't heard of anybody yet, within Accra anyway, who has been vaccinated and has developed clots. Prof.
Okay, thank you very much. There's a question. Is Dr. Bediakun with us? Dr. Bediakun,
Yes,
there's a question which I'd like you to answer. Yes, it says, can you have the Astrazeneca vaccine as a first vaccine and have a different type of vaccine, for example, the Moderna as the booster or second vaccine?
Okay, so that's actually a good question.
Is it advisable?
It's a good question because it's a reality that might in Ghana might affect us. Currently, I know there are trials. AstraZeneca is trying to do a trial. They are collaborating with the Gamaleya Institute in Russia to look about using AstraZeneca as the prime, as the first dose, and then Sputnik 5 as the second. Because these vaccines are quite similar. One is the chimp adeno, the other one is a human adeno. There is some thought that actually using the human adeno as the second, as the booster, might actually work well. So there is, if you go on the AstraZeneca, I think Gavi website described that this was a trial, this was a project that was being undertaken to actually evaluate using AstraZeneca and Sputnik 5 together. Now, the problem is the Moderna and Pfizer vaccines are really quite different. As we said, those are RNA vaccines, and I'm not sure how compatible. It's not so much side effects as you want to get the right immune response. So I really, I really don't think we have to worry. It's not like drug combinations where one drug mixes with another chemical and you have a problem. Vaccines don't really work that way. But the issue with vaccines is if you don't use them properly, then they don't work the way you want them to, and you don't get the protection. So currently what they're trying to do with AstraZeneca and Sputnik 5, which actually might be useful to Ghana, given the vaccines that are currently approved here are those 2. And I believe we are getting to the end of our first stock of AstraZeneca, and we don't yet know, we're supposed to receive more by May. So yes, there is work. Currently, no, there's no, no one is approving using mixing and matching vaccines, but there is work ongoing to at least as far as I'm aware of, to look at mixing and matching AstraZeneca and Sputnik 5. Now, I think there are some people who live in the US, got Pfizer, then came to Ghana. I think there have been questions about, can I then get, I suspect that if you got AstraZeneca, you'd have to get the second dose. So you probably, I really don't think there's any interaction. But in terms of the vaccines really working, they've been carefully dosed such that you typically need 2 doses of the same vaccine. So you can't really mix and match till we get evidence to the contrary. So I hope that, very long-winded answer, but I hope that gives some clarity. They're looking into it. Basically, we don't yet know if it'll work.
Okay. Thank you very much. That is that's a very good thing. Somebody else is asking, is it true that if you have had COVID, you need only 1 dose of the vaccine?
Well, that is also something that is being talked about. When you have limited stocks, people are always trying to see how you can make what you have stretch. So there is some thought that having one exposure, so having survived COVID, you basically had your prime, you've had your first vaccine, and then you could get a second one. Right now, the official stance in Ghana is everybody needs to get two, and I think that is based on the data that's available. Some countries have talked about using just, testing people and giving them one. I think we would only go to that if we just didn't have enough vaccine and you wanted to try to vaccinate as many people as possible, because it would involve testing people to see their seroprevalence, how much in terms of antibody levels they had against COVID before you decided they would be in the one vaccine arm. So for now, the answer is everybody should get 2 doses. But if the worst happens and the companies cannot match the demand and we're running out of vaccines, we may see an introduction of that kind of policy. But hopefully, vaccine supplies will continue to match the demand, and everybody can just get 2 doses and we can be done.
Thank you. So Dr. AKNK, I hope you're satisfied. He has a question for Radha. Radha, how many of your patients have developed diabetes after recovering from COVID? And has it reversed in those patients?
I haven't seen any. I haven't seen any. I've had patients who have diabetes that went pretty much out of control when they had COVID, and but some weeks after they recovered, most of them had to be switched onto insulin. But after recovery, things pretty much went back to normal. I haven't seen any who have developed diabetes as a
okay.
As
What
a
I can add
as a consequence.
As you said before, a lot of these patients, can I finish? A lot of these patients also may be developing their own chronic diseases. They may be along a certain path anyway. So they could very well develop it, which may not necessarily be related to the COVID infection. Okay, Bertha, go ahead.
Yeah, what I have seen in clinical
Bertha, go
practice.
ahead.
Oh, sorry. What I have seen is that because a lot of patients who have diabetes were one of the first people with risk factors or underlying diseases for COVID, there were a lot of people with new onset diabetes that were diagnosed who presented with COVID, not because they developed diabetes as a result of COVID, but because they had underlying diabetes and did not know, they presented with COVID and were told initially that they had diabetes. So of course, after the COVID disease has resolved, they still now have to start managing their diabetes. But I don't think the diabetes was due to COVID-19.
Great. Thank you very much, all of you so far. We've been so much. We've had questions. We've been able to really going fantastic. Is Dr. Menson, William Niaite. He's an MD with public health specialist, and he's with the vaccine program manager with the Clinton Health Access Initiative. He serves on the technical working group of COVID-19 vaccines in Nigeria, leading the capacity building work stream for CHI. And he's the lead fellow for health and equity at the Center for Social Justice. He was trained at KNUST Medical School and the Johns Hopkins University Bloomberg School of Public Health. So we're going Ghanaian international here. Dr. Manson, you have the floor. Thank you.
Thank you so much, Prof, for the introduction. We've been educated greatly by the panelists who went before me. And my part of the presentation will be in 2 parts. The first will be to dispel a number of myths that seem to be pervasive everywhere. And then if there's still more time, I will attempt to talk about the programmatic aspects of rolling out the COVID-19 vaccine from what I know is happening in Ghana and from my personal experience helping with this rollout in Nigeria. And so for the first part of this presentation, I will present a number of myths that exist, and hopefully by the time I'm done with this, a number of questions that our viewers would already have been answered. And so let's dive straight into it. So the very first myth that I will attempt to dispel is this, that the virus is just a mutated form of the common cold. This is patently false. This is the fact. The coronavirus is a large family of viruses that causes different diseases. SARS-CoV-2 does share similarities with other coronaviruses, 4 of which may cause the common cold. All 5 viruses have spiky projections on their surfaces and utilize spike proteins to infect host cells. And so from this, we know that the SARS-CoV virus is not a mutated form of the common cold. We now go to the second myth. And I think this has been addressed, and so I'll quickly read through the answer I have without elaborating too much. That the second one says that the virus was probably made in a lab. This is false. There's currently no evidence that suggests that this virus is man-made. And like I said earlier, SARS-CoV-2 closely resembles some other viruses that have triggered outbreaks in recent decades, but none on the scale that we have for COVID-19. And those two include the MERS virus, that's the Middle East respiratory virus. And then all the 3 viruses, there's a theory that they may have originated in bats, that they are zoonoses. In short, the characteristics of SARS-CoV-2 fall in line with what we know about these other 2 that made, a jump from animals to human beings. And so we do not have any evidence that this was made in a lab. The third myth is that if you have coronavirus, you will know. It is not necessarily true. And before I even read the facts as I have here, I'll use myself as an example. So full declaration, I tested positive for COVID-19 in October of 2020. And I was totally asymptomatic. I only found out because I needed to travel outside of Ghana for work. And so I went to Noguchi to do a test. And the following day, I picked up my results and it was positive. I was very confused, but I was reassured by the kind of stories I had heard from other people. And so I just went home, self-isolated. And fortunately for me, I did not get any symptoms. So I was finally able to get a negative test 2 weeks after that. And so you won't necessarily know if you have COVID-19. It causes a wide range of symptoms, many of which appear in other illnesses. And some of these symptoms have already been mentioned by Dr. Hackman, and so I would not need to go through this. But then we also need to know that a lot of people actually get infected, but do not show any symptoms. Now, another myth, which is quite popular, and I think one of our earlier presenters talked about it, vitamin C supplements will stop you from catching SARS-CoV-2. This is not true. And to borrow Dr. Yao's words, it's total hogwash. And researchers do not yet have any evidence that vitamin C supplements render anybody immune to COVID-19 infection. And you know that it may not even be enough to ward off the common cold. And so please be minded that vitamin C will not protect you from this. Another one is this, that antibacterial soap won't help against COVID-19 because it's a virus. So you know that before the virus was, before we had the vaccine, I beg your pardon, a lot of the preventive measures involved the use of sanitizers, hand washing, so that we do not keep these fomites and then transmit to the people we love. The truth here is that that actually works because the combination of soapy water and vigorous rubbing disassembles the virus building blocks and dissolves the sticky parts, which might cause the virus to adhere to human skin. And so hand sanitizers also work in a similar way. However, they are not as reliable as soap and water, which is why we always tell you to use hand sanitizers where you do not have running water and soap. Another myth, which did find expression in a lot of people's commentary a few months ago, is that you can self-treat by taking chloroquine. This is a lie. And although chloroquine and hydroxychloroquine have been mentioned as possible treatments for COVID-19, it's dangerous, it's lethal poison if not used correctly. If I remember a quote from one of the pharmacology lessons I did in medical school by somebody called Paracelsus, who, to paraphrase, tells us that every medicine is a poison. There is none that isn't. It is the dose or amount you take that differentiates a medicine from a poison. And so do not take this unless we finally have proof, scientific proof, that indeed hydroxychloroquine may be able to. But as of now, there's no evidence that it can be used to treat COVID-19. Well, somebody also says that we can't trust COVID-19 viruses because vaccines, I beg you, because they seem to have been rushed. And the vaccine had, this as mentioned by Dr. Bediako, and have been proven to be safe and effective. There are a number of reasons why we were able to develop these vaccines in such record time. It's because one, countries were able to prioritize people who were ready to be tested through the different stages in order to have us do this. And then thirdly, we were also able to leverage on some technology which helps us get, the mRNA viruses, as well as the one that's the DNA ones that get into cells using the adenovirus. And so they are safe and can be trusted. Somebody also goes like, we do not know what's in these vaccines. The fact is that for every vaccine, manufacturers need to publish ingredient lists. And in addition to the star ingredients, there are also adjuvants that help the vaccine stay longer and that help the vaccine be able to travel where it works. And so these vaccines do contain lipids and help deliver the mRNA into your cells. And despite theories, on YouTube, as well as some other social media, they do not contain microchips or any form of tracking device. Another myth is that I already had COVID-19, so I won't benefit from the vaccine. I think one of the panelists responded to this, and it's a fact that you can get COVID-19 more than once, but there are still many questions that remain unanswered. Day in, day out, we learn more about COVID-19. And so the current guidance is that even if you have had COVID-19 in the past, please go get the vaccine to enhance your own protection. And then one other myth is that since COVID-19 survival rate is so high, I do not have need for a vaccine. And this is false, and it's a very dangerous thought. It's true that most people who get COVID-19 are able to recover. It is also true that some people do develop severe complications, in addition to the older population, as well as those with underlying illnesses, whose outcomes may not be as good as the general population. Young, healthy people are known to have succumbed to this illness. And so please, if you do not want to become a part of that statistic, please go for the vaccine if you have access to it. So another myth is that once I get the vaccine, I do not have to wear a mask or social distance. Even if you get the vaccine, you should continue to wear a mask and practice all the precautions that have been preached to us. There are a number of reasons for this. And the first is that the vaccine does not, start protecting you the very minute you get it. It might take a few days after the immune response has come up before you get fully protected. And there's also another reason why we have boosters for these vaccines. Why for AstraZeneca, for instance, you do not just take one dose. And for those of us who have children or who remember how we were vaccinated when we were young, even for things like the polio vaccine and then the DPT and others, we needed to do it more than once. And if I remember, even for vaccines that are given in adults, for one like the hepatitis vaccine, after a 10-year period, you are advised to go for a booster. And so please, even after you get the vaccine, you will need to protect yourself and others. Some people will also tell you that they are not at risk for severe complications, and so they do not need the vaccine. Similar to an answer I gave a few seconds ago, you can still contract this, and people who are probably stronger and healthier than you have succumbed to this. And so we will need to get the vaccine. Oops. Right. Sorry. Right. So this is really the end of the dispelling myths part. I will use the next 3 minutes, and I'm not going to project, to talk about some of the thinking that goes into the programmatic aspects of rolling out the vaccine. And so to roll out the vaccine, it takes more than just getting the vaccine into the country and then administering it to people. And it's really a multidisciplinary venture that includes things like regulatory approvals, cold chain logistics, vaccine delivery strategy, monitoring, waste management, evaluation, advocacy, communication, and social mobilization, because we need people to know why they are taking the that we are using. And so we have segments in the virus, and then the second is your risk of morbidity, mortality or severe morbidity. And so we have 4 groups. The first has got to do with people who are most at risk. And so we have health workers, frontline security personnel, people with underlying medical conditions, people who are 60 and above, frontline members of the executive. And I think a part of the reason why we have to give some of our leaders before, even though they might not fall into these categories, is to assure the rest of the public that indeed the vaccine that's being given is safe and actually works. The second group has got to do with other essential service providers, including security agencies, teachers and students, people who work at fuel filling stations, air traffic and civil aviation control services, the police, and then the rest of the government. The third group is the rest of the general public that I did not describe earlier. So for now, it's all persons aged 18 years and above. And there's some thought about excluding pregnant women because during the tests, it may not have been tested on pregnant women. But then it's also known from the data we have now that some people who were pregnant actually took the vaccine and had good outcomes. They didn't know they were pregnant. And so these people are being followed to see if, anything might happen. And then the last group has got to do with group 4, all others. And this would be pregnant mothers and persons under 18 years. When we finally have enough knowledge to show us that indeed it is safe for this group of people. Thank you so much for your attention, and I'm happy to take any questions that you might have. Over to you, Prof.
Right. Thank you very much, Dr. Menson. That was a fantastic breaking or blowing of myths. I have a couple of questions that were WhatsApp me. COVID-19, initially, Dr. Asante said mainly respiratory, but this disease over the past year has shown us indeed that it is anything but just respiratory because it has affected virtually every single cell of the body. How is that explained? That's open to all of you, all the panelists.
So maybe I will make an attempt. So the virus prefers these really nice cell receptors. They are called ACE2 surface cell receptors. And these receptors are actually distributed in several places in the body. So you can find them in the upper respiratory tract, in the lower respiratory tract, in the gastrointestinal tract, so they are all over the place. This is how come the virus has also been isolated in stool, for example, because it can also go through your gut as well as the upper and the lower respiratory tract. And yeah, maybe there is some sort of dissemination as well. It could go into other organs as well. So this could explain why it's all over the place. It's all over the place, yeah.
Okay, thank you very much, Dr. Santi. Any other panelists would like to contribute to that? Then the other question is for Dr. Bediako. Dr. Bediako, you were talking about nanoparticles for the mRNA vaccines. And how many strands can fit in each particle? It's a question of, in each quantity of the vaccine, how many of these adenoviruses or these nanoparticles would there be in each quantification? And would it make any difference?
Oh, the quick answer is I don't know. I'd have to look it up. I should correct myself. The Novavax, I think, is going to use nanoparticles to package the subunits. The mRNA, I think, is liposome. So it gives a little, the RNA on its own, if you just put it and inject it, it would probably just dissociate. And you want it to get into a cell. So they surround it with a lipid membrane. Our cells have lipid membranes, and to cross a lipid membrane, you need to yourself actually be in a lipid. So the lipid will fuse with the lipid membrane and the RNA will come in. So in this case, so I misspoke. I think they are liposomes. So they are tiny droplets you could think of. When you add water, a good example, you add oil to water, you see those little droplets. These are, very, very microscopic versions of that. In terms of how many RNA copies are in each, that is something I'm sure we could easily look up. I'm sure it's there. I know the Astrazeneca vaccine, for the adenoviral vector, I think each dose contains a massive number. I mean, I can't remember off the top of my head. I have to look it up. But I remember when I saw it, I was like, no wonder I felt so bad. And it's because these are replication deficient viruses. So whatever you inject is all that's going to be there. And so you're trying, the immune, in order to, we have to remember, to protect ourselves, we have to charge up our immune system. We have to induce an immune response. So I will look up, I knew it, I think it's on the order of 1 × 10⁻10 or something, some ridiculously high number of adenoviral particles are in a single dose of the AstraZeneca vaccine. But it's been carefully optimized. It's not something to alarm anybody. You need to, our immune systems are responding to many things all the time. To get the immune system's attention, you have to hit it relatively hard. And that's what the vaccine is trying to do. But hit it hard without damaging the host, obviously. So in terms of how much RNA is in there, I don't know. We could look. But I think the more important thing is to recognize that whatever is in there, whatever quantity, has been carefully worked out to induce a good enough immune response to protect you, but is not going to, in any way harm you.
Okay, thank you. There's another question here. From one of my classmates, Dr. Kwame Boachi. I can see you are listening. It says, how true is it that people with blood group O do not get COVID-19 infection? Does anybody know? Is there any scientific evidence?
There is, I think there's a little bit of evidence at the beginning of the outbreak. I need to go back and review it more carefully, but there was some talk about the fact that people with blood group O, it's not that they cannot get it, they are not immune, but they appear to have a slightly lower risk. I haven't looked at the literature more recently to know whether or not that was borne out, but there was something about blood grouping, but it was not.
But then
it was not a complete immunity. I mean, people with blood group O have probably still died from COVID, but I think there was a relative, a small maybe level of resistance, but not much.
Yeah, I am blood group O. I got COVID, was COVID positive, tested 31st December 2020. Had it reasonably mildly because I didn't have to go into hospital, but got quite bad post-COVID with vertigo, labyrinthitis, and that has not been pleasant at all. So please, COVID is not to be joked with, not to be played with. It's a very serious condition. Can we just give the presenters a couple of minutes each? Bertha will handle this session just to say their last few words and to give their last advice. We've had a lot of comments and she will take that up as well. Bertha, over to you.
Okay, thank you very much, Prof. So we want to thank all the participants who tuned in, about 60 something of them, and those watching on YouTube, and also our wonderful presenters. So if you have any questions, there's still time to share it. In the meanwhile, while you're thinking of any questions, I would give each of the presenters an opportunity to share their last thoughts. And it says ladies first. So I'm going to
just one last question which has come in through the Facebook chat. Hopefar Addington says, please, if the side effects are seen within 24 to 48 hours after taking the vaccine, why are the pregnant women who have taken the vaccine still being followed?
Okay, well, thank you so much, Prof. Okay, so I would say a few things and then y'all can come in. Day in, day out, we are still learning about COVID-19 and we are still learning about the vaccine. What we know is that most of the effects, right, we call it adverse effects following immunization, do happen within a short period, and then appropriate guidance can be given. And so it's still a work in progress. Over to you, Dr. Widiafo.
I didn't catch everything you said, but I think the gist, all I was going to say was, we are, this is still a very new, it's a new virus and the vaccines are recently developed, even though they've gone through extensive testing. And so it's prudent just to continue to follow people up. In Ghana, they're not giving vaccines to pregnant women because, we are waiting to see, you rarely do trials in pregnant women. And so they typically are the most protected group. And so I guess for now, we're hoping that herd immunity will protect pregnant women. But, everybody is being followed up, even the people, even non-pregnant people, as far as I'm concerned, the FDA is basically on the lookout for any adverse events. There may be small populations of people with rare allergies or rare conditions that would be flagged. And so the idea is to try to identify these people. So I think it's just prudent to collect information, even if you don't expect there to be much problem. It makes sense to just be prudent and just be extra careful.
Dr. Asante?
So my last words?
Yes.
Okay. Yes. So what I would like to say is that, like we've all said, this is a new virus that we are still learning about. We still have a lot of things to learn with SARS-CoV-2, and it's a continuous learning process. And as the virus is going to go on transmitting and moving from one person to the other, it's going to change. Hopefully, most of these changes will not affect the larger properties of the virus. Please don't get COVID-19. Follow all the protocols that have been laid down. They are proving to work. As Dr. Hackman mentioned before, we have seen a few cases of reinfection. People have got COVID, cleared it, and then gotten it again. And usually on the second one, it's not a very good outcome. So please, please, please try not to get COVID-19. If you have the opportunity to take the vaccine, please do. When I took my vaccine, it completely knocked me off for 48 hours. But then I was then fine and then got up and came back. So kindly do not get COVID-19. Please take good care of yourselves. And yeah, thank you.
All right.
That's all I have to
Thank
say.
you. Wonderful. And before Dr. Hackman also gives her comments, there was a question in the chat about should you still wear your mask after getting the vaccine? And I would say a big yes to that, because just in the last 2 weeks alone, I have seen 2 patients fully immunized come in with full-blown COVID-19. Granted, they were all above the age of 70 years old, but they were fully immunized individuals. And if you look at all the clinical trials, even during the clinical trials, I can quote that for Pfizer. 9 people still developed severe COVID-19 who had received more than 2 doses of the vaccine. And don't forget, the Pfizer vaccine was supposed to be the most protective with over 95% protection, but some people developed COVID-19 disease. And the idea of the vaccine is not to prevent you from getting infection. All the vaccines have been shown to reduce the risk of hospitalization and death. And now we do know that you can get post-COVID long symptoms, including clots and strokes and whatnot. So if you can still keep on your mask, my children always tell me that I behave as though I haven't received the vaccine because I'm still, in fact, I've even forgotten I've received the vaccine. I still behave like I'm still protecting myself from COVID-19. So it's extremely important to make sure that you have your mask on all the time, or institute all the protocols that you had before you receive the vaccine. And someone also said, I understand one is only fully immunized 2 weeks after the immunization. And it's about 2 to 3 weeks when you build your full antibody response to the vaccine. But these people who came into the hospital had been immunized more than a month ago, but they still came in with COVID-19 disease with pneumonia. So I will really advise that until this pandemic is over, we thank you that you've received the vaccine or you're going to receive it, but please still be careful. So Dr. Hackman, if you're still there, kindly share your thoughts.
Yes, my final words. 2020 was a year like no other. I think we're all desperate to get back to normal. I've seen my friends in the UK, they've been under lockdown since December. We've worn masks. I haven't visited friends in a year. It's actually a year since I visited some of my friends. We want to go back to normal. Health workers are tired. It's been an exhausting year for health workers. I think we would want to do whatever it takes. Yeah, we've accepted that we're going to have to live with a virus, but we want to live with a virus, and have our lives back to normal as well. And I see the vaccine as the way out, to normalcy. So I would encourage those who haven't, gone vaccinated to have the opportunity. 3 districts of Ghana where phase 1 is going on. Vaccination is still ongoing, and your static vaccination centers. Look for them, and if you fit the group in phase 1 that's been vaccinated, please get vaccinated. Get vaccinated so we can all be together again. Just go back to normal. And the sooner we do this, the better, because new variants are popping up, all the time. So don't let the opportunity pass you by. Don't wait and see. People say, oh, I'm waiting for Sputnik because that has 95%, efficacy. Take whatever vaccine, you can get, because all vaccines have been proven to prevent severe disease and prevent death. What more do you want? What more do you want to see? Get vaxxed, please. Thank you.
All right, wonderful, Dr. Hackman. And Dr. Bediako, your final comments.
Yes, I'd like to mirror the comments of the previous of Dr. Santi and Dr. Hackman. Vaccination really presents us with the only solution to COVID-19. We have to maintain the protocols, as Dr. Aye has said. We cannot, even if you've received the first shot or even the second shot, you have to keep the protocols, because These vaccines are shown to prevent hospitalization. They do reduce the risk of transmission, but it's not perfect. And so you need to remember that there are people around you who have not yet been vaccinated. And so you could be an asymptomatic carrier. So you have to be careful. But, I think it's very important that we, it's good to be informed and it's good to look for information, but I think let's resist the urge to buy into every conspiracy theory that is out there. No one is out to try to get us. The world is facing a challenge that it has never seen before. In many ways, the whole world is in this together. And so I think we have to trust God, obviously, but also we have to work together to try to do this. Finally, what I would say is we cannot make the mistake we made at Christmas. We were highly irresponsible as a country in November and December, and we paid a terrible price for it. People have died who did not have to die because, quite frankly, we were foolish in December. And Easter is coming up.
Yes.
People are tired. We want to celebrate. Please, this is not the time to have big church services. This is not the time to have big Easter dinners. This is not the time to have big Easter parties. Let's maintain the protocols. Let's not find loopholes in our protocols and say, well, because in the church, it's fine. I find that, you understand the spirit of the rule. Super spreader events are events where people gather. Do not gather in large numbers, period, no matter what the cause, no matter what the reason. If we do that, we may be able to have a more normal Christmas. If we behave the way we did in December, Christmas will be canceled again, and more people will die who did not have to die. So that is the strongest thing I can say. Easter is coming. Let's not make the mistake we made in Christmas. Let's stick to the protocols, get vaccinated when we can, and look forward to a more normal Christmas, hopefully, or at least a more normal 2022. Thank you.
All right, wonderful. Thank you so much, Dr. Bediako. And the last person to give their comments is Dr. Manson. But before he does that, I have one question that came from Dr. Barbara Entwa. Maybe he can add that to his comments. Dr. Barbara Entwa belongs to the class of 1973. As a matter of fact, she's just authored a book on a woman's health. And so we're glad to have her join us. And her question was, is it true that Ghana is going to start vaccine production with help from or intelligence from the Cuban government? And what I have seen is that I know Professor Frimpong Boateng was put in charge of local vaccine manufacturing. And Africa as a whole is working together with Gavi, which is the Global Alliance for Vaccine Development, and a few other groups to seriously start looking at manufacturing vaccines on the continent. I know South Africa is set to produce some 300 million, but so far as the Cuba intelligence, I want to see if somebody has any specific comment to make. And I'll, Dr. Manson, if you have a response to that, you can share with Dr. Barbara Entwa, while we also congratulate her on authoring a book on women's health.
Thank you very much, Dr. Ayi. So quick response to that. I think Prof responded to that earlier, that we had reached out to the FDA. We don't have a response yet. And I've also heard these, but not from an official source. And whatever the case may be, investing in vaccine development is not something that will produce results within a 2 to 3 month period. It's an activity that requires a lot of investments, and that would take some time to fructify. And so it's really my hope that there's some truth in this, but I cannot confirm for now. So for my concluding comments, now there's no doubt that indeed COVID is real. And I'll quickly talk about vaccination. And if there's a single public health intervention that has saved the most lives, over the course of history, it is vaccination. And because of vaccination, we no longer have smallpox disease. Because of vaccination, polio is almost eliminated from the rest of the world. Because of vaccination, a lot of the diseases that caused your parents to be hospitalized no longer exist. Because of vaccination, a medical doctor like me, I've been a doctor for about 10 years, but I'm yet to see an actual measles case. I'll be able to recognize it when I see it because I've read so much about it in books. And if there's so much good news, it's because of vaccination. And now our mainstay for fighting this canker of COVID-19 is vaccination. And so please, whenever you get the opportunity, do not hesitate. And the earlier you get it, the better, because the vaccine is also fighting by producing new variants. And the earlier you get vaccinated, you cut, this evolution from, happening. And so thank you so much for the opportunity. I've had a lot of fun, and I hope we continue these conversations. Thank you very much.
Okay, wonderful. And just before I end, there are two people who've had their hands up, Dr. Yaobedi Akon and Dinah Issa. Do you still want to, do you have a question before we end the program?
No, I was just going to comment about.
Yeah, can I just say that I've seen a comment here which gladdens my heart and gives us a good reason for having held this webinar. Akosia Bafo Klu says she was very hesitant to go for the COVID-19 vaccination, but after taking part in this webinar, she's going to get vaccinated. It is so encouraging because, as you have heard, there's nothing secret or nothing hidden or nothing unusual. So very good. Sorry, yeah, please carry on.
Yeah, I was just going to comment that in terms of vaccine development in Ghana, I've been, so my director, Professor Wandere, has been asked to join a committee, and actually a few of us are in a technical working group to provide input into what it would take to develop vaccines in Ghana. So I know, at least that is what I know, that the discussions are beginning to take shape. I don't know anything about intelligence from Cuba or anything like that. But as Dr. Mensah said, this is not something that can be done overnight. This is really development for the next 10 years and will take significant investment from successive governments, maybe starting with the National Research Fund bill that has been languishing in Parliament for, I think, almost 4 years. So maybe now it will actually be passed. But yeah, this is a long game. It's not going to happen overnight. But hopefully
waiting for the next pandemic. Waiting for the
next
next
pandemic.
We'll
be ready for the next.
To just say about the Cuba issue, Barbara, I think Cuba is in the phase 2 or phase 3 of the development of their vaccine. So there were talks between the FDA. I believe it is into the approval process for us to get it. So as he says, developing a vaccine is not something that happens in a few months or overnight. So it's good news that we are about to start, but it will take a lot of investment. Thank you.
All right.
I think I heard on the news yesterday, actually, that the FDA, I heard Mimi Dako say that Ghana is going to go into the filling. So I think the vaccine will be brought to us in bulk, and we'll do the filling of the ampules and the labeling here. And that's due to start in the next 3 months. It was yesterday. I hope that helps, Barbara.
All right. Well, wonderful. Thank you very much. And on this note, I'd like to say a big thank you to Professor Ifa Hesse, the current president of the Old Girls Association of Wesley Girls High School. Wesley Girls High School is a wonderful school to attend. And like I said, a lot of us on this platform went to Wesley Girls High School. We appreciate all the girls' schools, but we love our school and we cannot hide it. Professor Hesse helped to put this together. The Wesley Girls High School Old Girls from the UK had one about 2 weeks ago, but we felt it was good to open this up for more people. So we want to thank her for her leadership. And I also want to thank the Old Girls Association webinar group. We came together last September to organize monthly webinars, and one thing kept on cropping up or another. And so in January, we thought, it would be great to put out one on vaccines. So we want to thank you all for attending and also thank the subgroup, and please be on the lookout for some monthly topic. It may not necessarily be health, but we hope to engage you in the future on different topics, usually using our own alumni to do this. And I also want to thank the speakers. It's usually easy to just log on and listen to someone, but you don't know the hours of work that go into preparing presentations, reviewing them, and just being available for 2.5 hours. So, because the speakers logged in 30 minutes before, and so they've been sitting here literally for 3 hours. So I want to thank every one of you. Dr. Radha Rakam, Hartman, you were excellent. Dr. Ivy Asante, the star of the 1997 year group, you were excellent. Dr. Yaubediako, thank you so much for availing yourself. And Dr. Manson, and we want to even thank all of you, and Professor Hesse too, for the fact that you dedicated yourself to study, to be who you are, so that at this very important time in history, your knowledge, your skills, your intellect becomes beneficial not only to your family, but to Wesley Girls and to the country at large. And to the participants, what can I say? 2 hours of listening to us change your minds. And I want to say this before I forget. I always tell this to my vaccine audience, that smallpox was the first disease to get a vaccine and the first disease to be eradicated with a vaccine. And you think that was enough? The United States is the only country in the world with 300 million doses of the smallpox vaccine available to immunize its whole population if there's an outbreak. And guess which 2 countries also have smallpox virus stashed away? It is the United States and Russia. If vaccines were this bad, US wouldn't stock 300 million doses. I have been vaccinated. If you joined us today and you're hesitating, go and get vaccinated and protect yourself. So on this note, I want to thank all of you. And it was a pleasure being a moderator for this event. And thank you all for joining us.
Thank you. Goodbye, everyone. Thank you so much, Yao and Dr. Manson and Ivy. Thank you. It's been fantastic. God bless.
Wonderful. And I'll leave the Zoom on for just about 5 minutes or so. Maybe somebody. All right. Thank you.